Hello, dear students. Welcome to my channel, Learn with Anshu. Today, I am going to discuss a new chapter regarding the molecular docking and its current scenario and its future prospects in drug discovery. See, the molecular docking is one of the molecular docking is currently a new scientific study regarding the drug designs, which is based on the computer programmings. So today, let's start the lecture. So molecular docking refers to a computational technique which is used in structure-based drug design to predict the binding of small molecules to macromolecular targets. It aids in studying biomolecular interactions and developing therapeutic agents by analyzing bond conformation and free energies of binding. So as you understand that basically any reactions which is based on the target protein and the ligand which is attached to it, or we can say small molecules. When it is studied on molecular level, it gives us a more clear picture on how the interaction takes place, and it helps the researchers and scientists to analyze the possible conformations, bindings, and helps to design the target sites and the ligands as per the structure. So we can say that the docking molecular refers to a computer-aided drug design technique used to predict the binding orientation of a ligand or drug in the active site of a target protein, providing insights into molecular interactions that can enhance the biological activity of the ligands through structure-based drug design. The molecular recognition process has been nicknamed molecular docking by analogy to a ship maneuvering in a harbor because it involves placing molecules in appropriate configurations to interact with a receptor. I hope many of you would be knowing the term the docking, especially when it is used to dock a ship at an appropriate at the seashore or at the port at a suitable site, or when we see in the space science that it is used to attach two important tools or dock the uh, two two parts of any satellites or rockets. So here, similarly, usually molecular docking, the correct term would be that basically it is used for the recognition of the molecules or to the modeling of the molecules in the way that it gives a correct configuration or hypothesize a better configuration or conformations in the structure of the molecules, and it helps to identify the possible target site. And molecular docking is a natural process which occurs within seconds in a cell. In molecular modeling, the term refers to the study of how two or more molecular structures fit together. Now here, this branch of science have prospered or flourished basically due to three important advancements in the science, that is the structural biology, then the high development or improved development or advancement in the computers, and the growing availability of protein 3D structures and compound databases. So what is basically the structural biology? So here and how it helps the in molecular docking. So what we see in the structural biology that it is a fundamental to molecular docking and providing essential atomic structures of target proteins, enzymes, or nucleic acids, typically generated by X-ray crystallography, NMR, or cryo-EM. It defines the active binding sites, analyzes conformational changes, and validates the docked poses, enabling accurate prediction of drug receptor interactions. For key roles of structural biology is that the structural determination and preparations, that is the high resolution structure from protein data bank are processed, that is removing water and adding hydrogens to create suitable receptor for docking. Then binding site identification. Structural studies identify binding pockets, catalytic residues, or druggable sites, that is the apo versus holo structures, which is critical for successful docking. Then modeling receptor flexibility. While docking often treats receptors as rigid, structural biology provide ensemble data to account for induced fit or conformational changes, improving accuracy. Basically, what happens that here the receptors, usually the docking structure is use treats receptors as rigid structures, but here it helps, the structural biology helps to determine, it also helps in determining the flexible part of the target proteins, so that it will help to induce to fit or conformational changes model can be added in that to improve its accuracy. Then validation of dog poses, insights from structures, that is the electrostatic hydrogen bonding allows researchers to interpret docking results and to determine if a pose is physically plausible. Then homology modeling, when experimental structures are unavailable, structural biology techniques, example is comparative modeling, are used to predict the 3D structures based on homologous proteins. So these are some of the important benefits of the structural biology, which helps in the analysis of any molecular structures of receptors and ligands. So here we can also say that without structural biology, it would be very difficult or impossible to analyze or interpret the the molecular interactions, or to design any molecular models. Here one thing is that that highly effective it is, helps also analyze it, or also gives uh, developing a 3D structures of proteins, which is basically very important, as it gives a high resolution structures of proteins, and easily it will help to identify the different orientations, or to predict the different orientations where the target, or where the ligand or small molecules can bind, or even we can say that the most effective site where the ligand can bind. Next, here the next important role is of the computer softwares. So here in the list, I have already discussed that some of the there are different docking tools that has been developed for both commercial and academic use, such as dock is there, autodock, flex, gold, glide, fred, moe dock, autodock, vina, which has been respectively developed by our renowned scientists, and it helps in designing the molecular model effectively and efficiently, and also it helps in analyzing those interactions in a better manner. And second, it stores different protein 3D structures in their software, and finally through those softwares we can take out, or we can find, or sort out some of the new structures or important structures, which will help not only in determining the orientations, but also the determine the correct atomic structures through which we can do the dock process. So how this software can help, different types of software can help in the molecular docking process. So first is that it is basically do through the data acquisition. So downloading receptors and ligand 3D structures, it can easily download them. And basically this type of softwares becomes very important, where it is not not possible through doing all those through experimental modeling. And also the softwares are quite effective, and also it saves time of the researchers and academicians, and these softwares are very effective. So if we compare from the experimental modeling and the computational technique, so it easily it takes shorter time, and also effectively manages the better understanding. And not only that, it also helps, the software also helps in storing the important data related to the receptors and target protein structures. So here, basically, first is that it helps downloads the all the important forms of data. Then again, it data pre-processing, that is the cleaning receptors and ligands molecules and saving them in protein data bank and CIF format respectively. And important energy minimization and converting molecules into autodock format. Now here one of the important thing, that the binding affinity, so it can be easily analyzed by the computer software, that what is the at the different poses or of the different interactions when there is done, what is the binding affinity, where is the highest binding affinity, and where is the minimum binding affinity. And that is based, and that is analyzed on the base of scoring. And on that scoring, we can easily understand or analyze that which is the best binding part of the target protein and the ligands. So here it helps into the energy minimization, and where the minimum energy is utilized, it is the best target site. Then it initiates the docking process, and it also saves the result. So basically here we can say that molecular docking is the study, it is a computational study of how two or more molecular structures fit together better, it how better and how in an efficient manner it can fit together. And it is used to predict how a protein interacts with small molecules. Now here, the next important is that it basically helps to, with these all those things, we can say that it basically helps in the understanding the behavior of small molecules in the binding pockets of target proteins, and can be described by molecular docking. Now here, every docking software program usually has a particular algorithm to identify the active site of protein by allowing binding of the ligand in different parts of the protein, and exploring the best possible binding positions of the ligands within the proteins, and reports an energy for the pose based on scoring formation. Docking can reliably tell what is very unlikely to bind, and good tool for compound library enrichment for virtual screening. So here this software helps in the virtual screening of these structures. And we can easily say through the image formations that which is the best binding structures. Now, these high resolution structures are processed to create suitable receptor for docking, and then it to predict, simulate, and analyze interactions between small molecules and target protein. Here, basically, if in concise manner, if I make you understand, so here, the three important functions of a computer software is that first is does the conformational sampling, then bind site analysis, and scoring compounds efficacy. So here, what is the term that is the virtual screening? You already know that it helps in the visibility. It increases the high resolution. It creates high resolution images of the 3D structures of the proteins. Second, in the conformational sampling, what happens that there is an algorithm which explore the possible orientation or poses of the ligand within the target binding fight sites, and scoring function is the calculate the binding affinity and explore the thermodynamic stability of the protein ligand complex to the rank of the best docked to the best dock poses and visualization analysis. Then this display the 3D structures. Next, that it was first developed by the scientist called Irwin Kunz of the University of California, who created the first docking program in the middle of the 1980s, and efforts are constantly being made to enhance docking computations. Irwin D. Kunz is an important figure in the field of computer-aided drug design and molecular modeling. He is a pioneer of the development and conception of the area of the study known as molecular docking. One of the first docking program, UCSF dock, was developed in his group in 1982. Dock uses geometric algorithm to predict the binding modes of small molecules. So molecular docking is a prominent method for structural analysis and designing of drugs, which is assisted by computer programs. The aim of docking is to predict the binding between two molecule ligand and receptor when they bind to form a stable complex. The ligand could be any micromolecule, but mainly this method is performed with protein of known 3D structures. Fruitful docking method utilizes a scoring function which appropriately ranks the docking candidates. It could be utilized for performing hypothetical screening of huge collection of diverse compounds which rank the results and proposes the hypothesis of process leading to ligand blockage to intent target which is not found appropriate for the process of lead optimization, which aims to increase the promising compound for enhancing its effectiveness, decreasing toxicity, and enhanced absorption. Besides all this, the setup of input structure for docking is as critical as the docking itself. The evaluation of the results of algorithm which could perform a broad search of design space and ignores local optima named stochastic search could also few times found to be unclear. As molecular docking has become important for drug design, so the reliable theories for this include sampling algorithms in scoring functions. And as you know, as already I have said that it plays an important role in a specialized area of drug discovery. It is a computational technique that predicts the optimal binding orientation and affinity of a small molecule to protein receptor, offering atomistic insights into molecular recognition. This method is a cost and time efficient procedure for drug design, particularly valuable for screening vast chemical libraries. Next important thing, that a good grasp of the principle of molecular recognition at the molecular level and the associated forces involved upon docking is essential to a good understanding of molecular function and biological process. Here you can see that understand how to understand it is very important that there should be a good hold on the different associated forces or different bonds in the binding involved upon the docking is essential to understand the molecular function. Here you can see that block the recognition, that is the impact, what is the impact on drug discovery. So to understand the molecular recognition, it is necessary that either these ligands that blocks the recognition, that is either they are inhibitors or antagonist, or it increases or emulates the recognition, that is the agonist or recombinant protein. So a good hold on the basic concepts of on the molecular recognition or the structures, to understand the molecular structures of protein and the molecular interaction or the bond formation between the proteins, either it is electrostatic bond, hydrogen bonds. So it is necessary that these structures should be well understood before going into the to for the molecular docking process. So these are the basics. So it is very important that we should be knowing about the protein structures or target structures or small molecules at its molecular level and how it will impact on the drug discovery. Next, what is the purpose of doing the molecular docking? So it predict binding modes of small molecule protein complexes, search databases of ligands for compounds that mimic the inhibitory binding interactions of an experimentally validated inhibitor, search database of ligands for compounds that bind a particular site of a specific protein, search databases of ligands for compounds that bind nucleic acid targets, examine possible binding orientation of protein-protein and protein-DNA complexes, help guide synthetic efforts by examining small molecules that are computationally derived, and many more. DOC may be used to explore the binding modes of an individual molecule or be used to screen a database of molecules to identify possible ligands. Now, here we can say that the receptor site is characterized by centers which may come from sphagen. Sphagen, that is the sphere generation, is a tool which is used in the UCSF dog that was developed by Irwin D. Kuntz and his colleagues suit for molecular docking to define the shape of a protein's binding site by generating sets of overlapping spheres. So basically, the UCSF doc is the basic or the basic tool for all the softwares. In short, we can say that all the softwares are inspired by the UCSF doc, which was developed by Irwin D. Kuntz. And here, what it does, it is based on that is the sphere generation, in which the molecular docking to define the shape of a protein's binding site by generating sets of overlapping spheres. So here what happens that there is a generation of overlapping spheres, and where these which is the best protein binding site. See, now various software has been developed which even discusses or which even evaluates the on genetic algorithm. So the software today, there are so many tools or softwares, and they have evolved on even on the genetic level. So the molecule being docked is characterized by ligand centers, which may be its non-hydrogen atoms or volume filling spheres called as sphagen. The ligand centers and receptor centers are matched based on comparison of ligand center and receptor center distances. Sets of ligand centers match sets of receptor centers if all the internal distances match within a value of distance tolerance. Here what we say that basically the reaction is basically between the ligands and the receptor centers which are present on the target sites. So here the ligand centers and receptor centers are matched based on comparison of ligand center and receptor center distances. Sets of ligand centers matches. So there are we already know that in there is a PDB, that is a protein data bank, which stores all different type of structural proteins in itself, and from those software and from those datas we take out the suitable structures which will be best possible structure or in a best manner which can attach to the target proteins or the receptors. So here sets of ligand center match set of receptor centers if all the internal distances match within a value of distance tolerance. Ligand receptors pairs are added to the set until at least nodes minimum pairs have been found. At least three pairs must be found to uniquely determine a rotation or translation matrix that will orient the ligand to the receptor site. So here at least three pairs must be found. So three pairs are very necessary to find uniquely determined rotation or translational matrix that will orient the ligand in the receptor site. Molecular docking predicts the preferred orientation and binding affinity of a ligand to a target protein. It works by sampling ligand conformations in the binding site and scoring them based on thermodynamic stability. That is the lowest energy and complementarity. Core principles include rigid or flexible docking, energy minimization, and force field based scoring aiming to simulate non-covalent interactions like hydrogen. So here you can see through structure that what is the rigid structure and what is the flexible. Here already we have discussed that what is the rigid, one is the rigid flexible docking. Then second is the energy minimization and force field based scoring. So here you can see that what is the rigid docking. Rigid docking is nothing but only the here the receptor is not, it does not change as per the ligand. So here only this ligand will set on that particular part of the receptor where it is best attached. Whereas in the reflexible binding, there is the receptor have a conformational changes or ligand have a conformational changes as per the receptor binding sites. If in more detail, if we can understand that in the rigid ligand docking, the most basic type of docking method which treats both the receptors and ligand molecules at conformationally rigid means that it searches there is no changes in the conformation, only the best possible site where both can attach together, and it searches for rigid body transformation that best fit the ligand into the receptor. Whereas in the flexible ligand docking treats the ligand as a conformationally flexible molecule. Means here that ligand can change its shape. See here, you can see that ligand is in this form, but seeing the receptor structure, it changes its conformation so that it can attach to the receptor part. So here you can say that flexible treats the ligand as a conformationally flexible molecule by searching over both ligand and conformations and rigid body transformations to identify the best fit of the ligand in the receptor, which is treated as a rigid body. So methods, docking methods are used to place small molecules in the active region of the enzyme. In addition to these methods, scoring functions are used to estimate a compound's biological activity by looking at how it interacts with prospective targets. Molecular docking is considered to be the most widely utilized computational phenomenon in the field of computer-aided drug design. Here you can see that one more thing, that is the one flow possibility and another is another flow possibility. So what is one flow possibility, which is given by the green arrow? So here it is the PDB files. Here the moleculars on the molecular surface, that it determines the critical features, and finally the geometric modeling is done, and after that verification and geometric scoring, that energy calculation and active site rotations and translational possibilities is been predicted. Whereas another flow possibility, what it happens that here there is some another inputs in addition in relation to the PDB files, there is some other inputs through which the molecular surface, through which the ligands are attached to the molecular surface. And here what happens, these molecular surface then focuses on the active sites, and after that again it goes to the geometric modeling. So here critical feature part is missing there, and again it focuses, and then it goes for the geometric modeling, then verification and geometric scoring, energy calculations, then active site rotation and translation possibilities. So a typical comprehensive workflow for molecular docking is done through two ways. That is the one flow possibility, and second is the another flow plus possibility. So what happens that in comprehensive workflow of molecular docking, used in docking is used in structure-based drug design to predict how a ligand binds to a receptor or target protein, follows a structured sequence from preparation to analysis. So molecular docking traditionally focused on docking a flexible ligand into a rigid protein has evolved to include several alternative workflows. That is the another flow plus possibility, and that improve accuracy and efficiency by accounting for molecular flexibility and using advanced sampling techniques. So here you can say that now so much advancement has been made that these structures not only can be taken from PDB files, but also there are some other inputs through which they can be derived, or they can be taken out. So next, here, basically, if you'd be knowing that is the enzyme substrate complex. So here, that is the model on which the molecular docking is based. So here, first, the model was that is the lock and key model, which was given in 1890 by Emil Fischer. And you can easily see that the best, which is the right key, which is the ligand part, which can fit into the target protein, and then only the enzyme substrate complex is formed. So a substrate is inserted into active site of a micromolecule in the same way as a key is inserted into the lock. So biological locks exhibit distinct stereochemical properties that are required for their operations. Next, you can see here that is the induced fit theory, which was given in 1958 by Daniel Koshland. And the fundamental concept is that throughout the character recognition, both the ligand and target adapt to another by modest conformational changes until an ideal match is reached. So, as we have seen that in the flexible docking, the ligand makes conformational changes as per the requirement, that so that it can best fit into the receptor part. Similarly, here also that same based on that principle only, that was given in by Daniel Kostland, that induced fit theory, that both the substrate and the enzyme both make conformational changes so that they can adapt to one another by modest conformational changes until an ideal match is reached. Third model is also has been devised, that is the conformation ensemble model. So in addition to small induced fit adaptation, it has been observed that proteins can undergo much larger conformational changes. A recent model describes proteins as a pre-existing ensemble of conformational states. The plasticity of the protein allows it to switch from one state to another. If I can show you here, see, this is the induced fit model, and this is the lock and key model, which is the rigid model. And here it is it was given in 2003. That was the here the new conformation ensemble model was given here. You can see here that how these are the this is the protein target structures, and these are the receptor molecules which attach. These are the target proteins, ligand molecules, which are multiple targets they are recognizing here. So here you can see that the initial condition for conducting a docking screen is the sequence of a specific protein. The structure is often discovered using a biophysical approach. So the structure of these proteins are basically discovered through the biophysical approach like X-ray crystallography, or less frequently NMR spectroscopy. A docking tool uses this protein function and a database of compound as inputs. A docking program success is contingent upon three sections: the search algorithm and the scoring mechanisms. While searching the conformational spaces of a protein bound to a ligand, the search space encompasses all potential directions and conformations of the protein. With current processing resources, it is difficult to exhaustively traverse the search space, which would include enumerating all potential molecular distortions and all potential translational and rotational configurations of the ligand reference to the protein at a moderate criterion of resolution. The majority of docking systems in use consider flexible ligands. Other attempt to represent a dynamic protein receptors. So these are some of the protein models on which the mechanism of docking is based. And on these structures, we can say that best possible target site, it helps in orienting the best possible target site where the ligand can attach. And with this process, since it is a very sophisticated process, and it is done on a very molecular level, so it is becomes a very important tool to which helps in the computer aided drug design. And for this it is necessary that we should be this is necessary that we should understand the molecular structures of different proteins, as well as the correct ligands, or the correct the ligands or small molecules which can attach to it. So this is a new tool, and it is a very promising tool or advanced tool which can help in identifying different type of drugs, and it will help to even to the treat the important the current scenario of multi-drug resistance pattern, which poses a big threat to the human population. So with the help of molecular docking, these MDR strains can be properly treated, and the this MDR the issue can also be overcome with the help of molecular docking. And as we see that it is a computational technique, so we can get more precise and effective methods to model the correct protein and ligand structures, and therefore it is one of the most important tool in computer aided drug design. With this, I conclude my lecture, and I hope it would have been very informative to all of you. Please do like and subscribe my channel. Thank you.